Bozeman, Montana. My name is Angie Weichert. I'm the Director of Education here. I'm going to be your MC for our live streaming event today. A big thank you to Streamable Learning for hosting this for us, and we're so excited that all of you are here joining us. Before I pass this over to our two scientists that are going to do our presentation today, I wanted to give you an idea of where we're at. So you may have not been to Montana. What I'm going to do is hop off this, this video feed, put you back on our computer screen, and zoom in on a map so you can see where we're at today. So give me just a second, and I'm going to jump on in here. All right, so there's a map of the United States. We've got California and Alabama joining us today. So we're north of you both. We're up in Montana, which is up along the Canadian border there in the northern Rockies. We zoom in to our state of Montana. You'll see that we're just north of Yellowstone National Park. Now, the Museum of the Rockies does not look like this today. It may be sunny and warm where you guys are, but for us, there's quite a bit of snow outside. We've had a big snowy winter. But our dinosaur out there gives you an indication of what's inside our museum. So while I lead you through a quick tour of our dinosaur halls, go ahead and type into that chat box if you can guess what dinosaur is greeting you outside of the Museum of the Rockies. This dinosaur's name is Big Mike. Can you tell me what kind of dinosaur Big Mike is? While you think about it, I'm going to keep going. Inside our museum, we have the Siebel Dinosaur Complex, home to our Dinosaurs Under the Big Sky exhibit. Within that exhibit, we have a whole bunch of dinosaurs on display, both real fossils and replicas. It starts off here in our hallway of, or in our Hall of Giants. And I see that both of you guys jumped in and let us know that you think that's a T-Rex, and you're absolutely correct. Museum of the Rockies is home to one of the largest collections of T-Rexes in the world, and there's one of them inside of our halls. That's Montana's T-Rex. It's a different T-Rex than the one that the cast outside, but this one that's up in our dinosaur halls, those are real fossils on display. We also have one of the largest collections of Triceratops in the world too, and so one of the cool things about the Museum of the Rockies is that we have enough fossils, enough specimens, or enough dinosaurs that we can put a whole sequence of dinosaurs in a row to show how dinosaurs grew over time. And what we're going to be talking about today is how we know different things about how dinosaurs grew and behaved based on not the outside of the bones, but the inside. So with that, I'm going to turn this video back on and introduce you to one of our scientists today. So we are now underneath the exhibits that we just showed you. So we're sitting underneath the dinosaur fossils that are above us. And I have two very special scientists here in our Histology Lab, and you'll learn what that word means in just a few minutes. But first, I'm going to introduce you to Dr. John Scannella. He's our curator of paleontology. Thanks for joining us, Dr. Scannella.
Thanks, Angie. Hi, everybody. So every summer, we go out into the field outside and try to find new fossils of dinosaurs and other extinct creatures. And if you ever go out looking for dinosaur fossils, sometimes when you find them, they can be broken up into little tiny pieces. They can be very delicate, fragile. And so it's very important to protect the fossils. Once you find them and you dig them up, you have to protect them so you can bring them back here to the museum where we can study them. And the way we do that often is with something like this. This is called a field jacket. So there's a dinosaur bone inside here, and this is layers of burlap and plaster that have been wrapped around this bone to protect it in a similar way to if you were to break your arm. A doctor might put a cast around your arm to protect the bone. So we protect the dinosaur bones in a similar way and then bring them back here to the museum. And then once they get here to the museum, fossil preparators are people here in the lab who work on removing all the rock from around the fossils so that we can see what they look like. And so they'll open a jacket, remove the rock from around it. If the bone is broken or in pieces, they might glue it back together. And then once they've prepared the fossil, we might have something that looks like this. So this is the horn of a little Triceratops. And so this is where the eye of the Triceratops would be, and the front of the face would be that way. And so by studying lots and lots of Triceratops of different sizes, it was discovered that when you find little Triceratops like this, the horns above the eyes curve backwards. So the front of the face is that way, and the horn is curving back towards the back of the skull. And so we can learn a lot about dinosaurs by looking at the outsides of their bones and see how, for example, how they might have changed as they grew up from babies to adults. So over here, this is another example of a Triceratops horn. So in this one here, the front of the face would have been this way, and so the horn is again curving backwards, which means this is a juvenile Triceratops, a little Triceratops. But you might notice that this horn looks a little different because it's cut into pieces. And so the reason it's cut into pieces is that we can learn a lot from looking at the outsides of the bones of Triceratops and other dinosaurs, but sometimes we can learn even more by looking inside the bones of dinosaurs at the microscopic level. And so here at the museum, when we need to study dinosaurs at the microscopic level, we bring them down here to the Histology Lab, where they can be processed by the Histology Lab manager, Ellen Lamb.
Great. Excellent.
Right.
All right.
Hi, Ellen.
Hi, everyone. Thank you, John. So I am Ellen Therese Lamb. I am the Histology Lab manager here at the Museum of the Rockies in our paleontology department. And I'm also sometimes known as the chief bone chopper. And you'll find out why in just a little bit. Today I'm going to teach you about paleohistology, which is the study of the microscopic structure of fossil specimens. And we do this work, like John said, we want to know more about the outside of a dinosaur. So the way to find that out is actually to go inside of its bones and look more carefully. We have a variety of different kinds of specimens that we examine, and we ultimately produce thin section slides to do this research. So today I'm going to show you the kinds of specimens that we examine, not only dinosaur bones, but also different fossil specimens as well as some modern ones. I'm going to show you the process of paleontology, how we actually get inside of a dinosaur bone on a small scale and then actually on a very large scale. Then we're going to watch me work on some of the different equipment to do some of the different steps of the process. I'm going to show you some projects that we do in paleohistology, approaching it from what kind of questions that we asked and then what kind of answers and evidence we found out about that outside of the dinosaur by looking inside of its bones. And then at the end, you're going to get an opportunity to see how a dinosaur bone matured from a very baby, small hatchling dinosaur right out of the egg all the way up to an adult. So we'll do that by looking again inside of its bones. So right here, to begin with, I want to show you some of the variety of specimens that we look at. So in addition to dinosaur bones, this is a Tyrannosaurus rex rib. And we'll come back to that later and learn quite a lot about this particular specimen. We also examine very delicate ossified tendons.
So, Ellen, could you, since we have a younger age group today, could you give us a brief description of what you mean by ossified tendon?
Right. Okay, so this structure within a dinosaur bone, actually, it's another large word called ligament, and it's an important word to know. They function as ligaments within dinosaurs. So this piece of connective tissue actually forms and then hardens as the animal matures and connects all of the bones of its spine together and create the posture that we now understand dinosaurs have. So you may have seen older pictures of dinosaurs in older books or maybe in some exhibits at different museums where the dinosaurs are depicted dragging their tails on the ground. So understanding how these ossified tendons work, much like holding up a bridge, will enable the dinosaurs to hold their tails up off of the ground. So that's this structure, and we're going to look at that in a little bit underneath the microscope. We also look at dinosaur teeth. So this tooth is from also, like Big Mike that you saw in the beginning, a T-Rex. And this smaller tooth is from an Allosaurus, another carnivorous dinosaur. So we look at these ossified tendons, we look at teeth, and we also look at dinosaur eggshell, which is another specimen that preserves well. Try to hold it up. It's very small, but even a small fragment like this can let you know a lot about the animal that left it behind. So you see the scale of how far we go. We start with this full specimen, and then we end up with these very, very small, in some cases thin section slides. So are people able to see that? Excellent. Okay, very good. So this is one of those ossified tendons. This is a very small dinosaur tooth.
So you put it right in front of your black shirt there a little bit.
Would that work?
It's hard to see.
It's hard to see. Okay. And then this is a very thin slice of dinosaur eggshell. How about if I do that? There you go. Now you can see some light going through it. So primarily we look at dinosaurs, and primarily we look at dinosaur bone, but these are some other kinds of specimens we examine as well. And we also like to compare extinct animals like dinosaurs with modern animals that are living today. So sometimes we'll examine modern specimens. So two of them I have right here to show you. One is an alligator femur or upper leg bone. So we might also look at a specimen like this to compare and contrast with dinosaur bone structure. And then something else that we might look at too, either the actual animal or this eggshell, this is from an emu. Okay, so I'm going to tuck those away. And now our next step is to take a closer look at some of these specimens underneath the microscope. So Angie's going to switch over and I'll describe to you what you're seeing.
Okay, you got it, Ellen.
Okay. So what age group is everybody
We're
out there?
preschool through 5th grade. Preschool,
through
2nd, 4th, and 5th. That's
Okay.
what we've got.
Okay. So that is what an ossified tendon. So you guys are going to get a lot of terminology and a lot of words that maybe no other elementary kids are hearing today on the planet. So I'm going to repeat them over and over. But the exciting thing is, once you've heard it, you've had it described to you, and you've been able to see it within dinosaur bones. So if you're interested in studying this any day, this will all sort of start to become familiar. So these ossified tendons actually functioned, as I said, as something called a ligament, attaching bone to bone along the whole backbone or spine of a dinosaur. So this helps us understand dinosaur posture better. So when we build an exhibit and we put a dinosaur in 3 dimensions back together, we want to get that posture right. So the next slide. So this will show you dinosaur eggshell, and we can actually study what are called the gas exchange pores. So that's where gas and pretty much air and moisture transferred between the outside and the inside of the egg while the animal was developing. So we can study the structure of those pores and then the mineral, the actual eggshell left behind, and that can let us know what family group we have. Because sometimes at a nesting locality, you're going to have little tiny baby bones and you're going to have eggshell, and it's really hard to figure out exactly what dinosaur this was. So the eggshell can help us determine. And now teeth. So on the screen, hopefully you can see lots of lines. And those are called lines of von Ebner for the person who originally discovered them. And what we also call them are daily lines. And each of those lines and the space in between represents a day in the life of the tooth. Because dinosaurs grew and replaced their teeth throughout their lifetime. So we get 2 sets. Sometimes I wish we had more. You may not wish that now, but you may wish that when you're older. But dinosaurs constantly grew and replaced their teeth. So we can actually chart the age and the shedding rate of the individual teeth by counting these lines underneath the microscope. Okay. So before we get back to bones, I'm going to have Angie move back and forth between a slide. So that's a T-Rex bone. That's a mature adult T-Rex bone. She's going to go back and forth, and you can see these marvelous colors develop. So the first one that's sort of tan, those colors are made by the minerals in the soil that get absorbed into the bone as the bone fossilizes. But the next picture, do you guys have any guesses how we get all of those marvelous colors in our dinosaur bone?
This is good. You already got a question that asked,
okay.
Did you use stain on the slide of the tooth, or is that the natural color?
Okay, excellent. Wonderful. You guys are on it and paying really good attention. Stains are exactly what are used with modern bone. But you need actual biological tissue to be retained and intact, exactly like it was when the animal was alive, for those stains to bind and stay attached to your specimen. So generally, we can't stain dinosaur bones. Here, what we use is a condition called polarized light that we create under a polarizing microscope. And we're going to show you on the next slide. Excellent. So this video is available, and your teacher can help you connect to this on the home page of our Histology Lab website. And this will describe to you exactly how polarized light works and give you some really good visuals, how the light goes through the bottom of the microscope, how the light waves are oriented depending on something called a polarizer and an analyzer, and then how when you introduce the specimen into that wave of light, how whatever kind of specimen that you put there changes the wavelength. So when you meet it at the top of the microscope with either the camera or while you're peering through the microscope, the light colors will change. And that depends on what kind of mineral the specimen is made of, and in the case of bone, how what are called the collagen or the protein fibers are organized in the bone. Okay, so one more bone picture on the next slide. And any other questions before we move back to bone? Okay, so quickly on this slide, you're going to see this slide or something similar to it. So this is again mature dinosaur bone. And again, you're probably some of the only kids on the planet looking at this today, which is pretty exciting. And I want you to start thinking about what some of those shapes and some of those structures may be, and it's actually very similar to what you'd find in an adult or what your teacher's bone may look like. Yours is still growing, so it'll look a little bit different. But on the left side, you've got some open spaces, and those are called vascular canals, and that's where the blood gets into the bone to keep the cells that are in there alive. And on the right side, you're going to see a whole bunch of lines in a curved shape. So get a chance to look at those and know we're going to come back and describe those more in detail in just a bit. Okay, so now we're going to move on to the process.
You ready to go back to you?
Ready to go back to me.
There you are.
Okay, great. Okay, so we're going to come back to this Tyrannosaurus Rex rib. And should I hold things up, or do you want to pan down?
I think holding them up would probably be better if you can pan them all.
Okay, very good. So this project shows you, woo, there's the lab. There's lots of pictures.
I think I hit the button that made it go to center.
Okay, very good. And I'm looking for my little quiz A. It's just disappeared. So John, I'm going to ask you to make, while you're waiting, a little substitute A we can use for this quiz. So just put a little the letter A on a piece of paper.
Okay.
Great, thanks. I don't know where it went. It just went flying off the book. Okay, so this project here shows you many of the pieces and parts that go along with the paleohistology project. So one thing we want to think about, you may have heard this before, is to put it back the way you found it. So before we start cutting a bone up We have to begin with a question in mind, so we know it's important that we do this work. And then once we have our question in mind, we need to decide how to go about it, and then how we're going to get the bone back together in one piece when we're finished with the work. So sometimes we'll make a replica of the entire bone, and the replica will be called the cast, or sometimes we'll just take a little segment out of the bone, replicate that, put it back with the original bone, and glue all of the pieces back together. That's what we did in the case of this T-Rex rib. So now this bone can go back to collections and can be studied. So we'll take the original bone and we'll put it in something that we produce called a mold. And this one is made out of putty. And then once we take the original bone out, we can fill this mold with plastic resin and make a cast. So this is an exact replica of the original piece of bone that we took out of the rib. So the cast will get painted to look like the original bone, and everything will get glued back together. The actual fossil will then get put into what's called plastic resin. Just like that. So this starts out as a liquid. We submerge the bone in the liquid, and then we add a special chemical that hardens it. So you may want to think about, why would we do that? Thank you, John. That's for our quiz later. So that's something to consider. Why would we put the bone inside of this resin? The next stage is cutting a wafer of embedded bone. And then we'll mount that wafer down onto a glass slide using what's called epoxy glue. And then we're going to spend a lot of time at something called a grinder polisher. So we're going to go over there a little bit later in our session this morning and show you how that works. But essentially, the grinder is going to enable us to remove almost all of this material, and then the polisher is going to help us get all the scratches out at the end of the process. So again, we end up with a very, very thin slice of bone, and I'm going to hold it up and bring the light around here like that. So now it's thin enough for light to be transmitted through the specimen and for the structure inside to be revealed. So any guesses from the group how thin this may be when it's finished?
Maybe this group could give you something that would be about the same thickness.
What would be about the same thickness?
Maybe you could tilt it again so we can see the side of it.
And it's something we're all very familiar with.
How thick do y'all think that is? Is there something that you could compare it to?
Knowing that we started here and we finished up here. So I'll let you know, because it's something you all have access to right now. When we're finished, it's about as thin as a human hair when we're finished. And the actual measurement is called about 100 microns. So very, very small. It's just a small fraction of a millimeter, which is very small as well. So that's our finished thin section slide. Great. So I'm going to apply the A back to our specimen. Excellent. And we'll check here. Any questions from everyone yet?
So far, if you've got any questions, go ahead and type them into that chat box and I'll read them out loud for Ellen.
So if not a question, then I want you to pay good attention to this bone. And now I'm going to flip it over and we're going to have it as a quiz in a little bit. And John's going to help us make a transfer of materials. Okay. I'll hold this up while John comes in. Okay, now we're going to get the next specimen repositioned. And if you can pan back out, there we go. So this is the other end of the spectrum in terms of size of specimens. This is a T-Rex tibia or lower leg bone. And again, the goal was to start here and then end up here. This is what the inside of a T-Rex bone looks like. This information will let us know a lot, again, about the age of the animal in this case, which is what we were trying to determine. So I'm going to point out some structures, and I want you to think about them. These here are called lines of arrested growth, or lags for short, and they might be similar to something that you've seen out in nature. So I want you to give that some thought and think about how counting these lines can let us know something really important about this fossil specimen. And again, even though you guys are younger, I'm going to give you these words because these are good and valuable terms to know in bone biology. So the vascular canals are these open white spaces, and again, that's where the blood supply comes into the bone and keeps the cells, which are actually alive during our life and embedded in all of the bony matrix. That's how the bones stay vital and alive, bathed in blood and tissue fluid. And then these little circular structures are called osteons. And what I want you to imagine is this slice was taken out like this from the dinosaur. And these circles of bones are actually long columns of structure that are running all the way through the length of the bone. So we're seeing it in what's called 2 dimensions, but in life you have to remember this is all happening in 3 dimensions. And these would be similar to like a long piece of licorice with an opening in the middle, or a long straw that was very thick in its perimeter. And this is pretty much how bone matures and then grows and develops, and actually what we call remodels throughout our lifetime.
So you've got a great guest coming in from California, from our preschooler, 2nd grader, and 5th grader.
Okay, so we'll go back to our quiz, or for this?
From that picture.
Tell us about these.
And you asked what those lines reminded them of, and the response out of California from those 3 kids were tree rings.
Tree rings. You guys got it. Excellent. So similar to tree rings that chart the annual development of a tree, each line will represent a year in the life of the tree. The space between is similar in bones. So we have lines of arrested growth, and all of the area between each of those lines represents a year in the life of the dinosaur and how much bony matrix was laid down between lines. And at these lines, bone growth slows or actually ceases entirely and then begins again annually. So we can count these lines and figure out the chronological age of the animal. However, you can start to see in here, and you'll see later in the presentation, that once bone begins to remodel and these structures form, sometimes they make it really difficult to read those lines of arrested growth. So that's a challenge that we have in paleohistology right now, figuring out those younger years in an older animal. So great guess. Excellent work. Okay, so on this tray, we have a number of pieces and parts from this paleohistology project. So again, we made a cast replica, and in this case we made 2. One was used to restore the fossil, and the other one was made so a researcher who's also working on this project can study the actual specimen, study the structure of the bone, and because this specimen is so large and I can't make a thin section that big, I actually have to divide this bone into 4 pieces. So this cast will help them put all of those pieces back together. This is the mold that we used to make this larger cast, and this is a different kind of mold, but it still uses a silicone-like compound that goes in 2 parts around the original fossil, and then we make what's called a clam shell on top, and this is made of plaster like you saw with John's field jacket, and then fiberglass to help that silicone hold its shape around the fossil. So the 2 parts are put together. I can do this one hand. Okay, let's close. And clamp together tightly, and then this opening here allows us to pour in casting material and make a replica. So as you can see, we get to work with a lot of really fun materials as grown-ups, which is great. Okay, so more parts and pieces to this project. Again, the bone is embedded in plastic resin. Any answers on why that might be necessary?
So think about, we asked this earlier, but I didn't tell you to type it into the chat box. If you have a guess as to why we put all that plastic around that fossil, type that into the chat box so I can tell Ellen. Why so much plastic? Why do we put it in all of that resin? I'll zoom into it here. There we go.
Okay, so we have the embedded bone, we have the slice, and we made this slice on a very large tile saw with a special blade that actually has diamond around the edge of the blade, hardest substance, and that is what we need to cut through dinosaur bone.
Right, so we've got an answer.
We have an answer.
Oh, both of you answered. Fantastic. So one answer is so it doesn't break when you try to cut it, and then the other one is it's fragile.
Okay, you're all hired. That's exactly what we need to do. So we need to be very careful with these precious fossils during the process and make sure we have minimal damage to the specimen. And then once we get our piece extracted for study, exactly. We already have to cut this in 4 pieces. We don't want it to break apart anymore. So that's exactly correct. Embedding enables us to cut this nice thin wafer. That wafer gets mounted to this slide. So that's to a large glass plate. And then again, in a little bit, you'll see me on the grinder working with essentially a very, very rough sandpaper to go from here to here. Okay. So next I'd like to share with you 2 parts of the paleohistology process. So first you're going to watch a video of me cutting open a dinosaur bone, and take note of the safety gear. And Angie's going to switch over to that. And then we're going to come back into the lab and I'll actually be on the grinder and I'll show you a little bit about the grinding and polishing process, and you can ask me some questions.
Okay, there you go.
Oh, we didn't do the quiz. Oh, that's right. So I'll have to do the quiz before the grinder. Thanks. Cue, John.
All right. You're back to you.
Okay. Did we do the grinder?
Oh, I thought you wanted to do the quiz. I'm sorry. I zoomed back out.
Oh, we can, right. We can do the quiz and then we'll do the grinder?
Yes.
Perfect.
We're
Okay.
back on you. Quiz
Okay,
time.
so we'll do the quiz. Very good. Okay, so we've got this large tibia, and then I'm going to pick up and check that you can see. Excellent. The smaller TRex rib, and everything's labeled with letters. So your challenge is to see what part of these fossils are real and what part are cast pieces or replicas made to restore the fossil. So here we've got section A, B, C, or D in the rib.
So you're going to type into the chat box right now one of those 4 letters. Which part of this rib is a replica? What's the piece that we put back in to replace the one we cut up? Can you guess? A, B, C, or D?
Okay.
Which one do you think is the replica? Oh, we got to answer. I'm going to wait one more. We'll see if Shauna wants to jump in here too.
Okay.
There you go. They both answered C.
Okay, very good. Excellent work. And you might have seen that cast earlier and noticed the shape, so good work. And I'm going to turn this over and show you what we have here is a yellow mark, and that is what we call our secret code. Anytime we return a specimen to collections with part of it being a cast, we mark a yellow band on the cast piece. So 100 years from now, when someone wants to study this rib again, they'll know that this part is a replica and the rest is real. Okay, so how about in this tibia? We've got A from the end to here, we have B, we have C, and then D.
So same thing, type on into that chat box. Which one of those 4 areas do you think is the replica? A, B, C, or D? So maybe you could point those out for us one more time.
Okay.
A.
I'll show you A, B, C, or D. And then I'm also going to take out some pictures to show you why all of those letters are so important.
So we have a guess of B and also C and B.
B and C. Ah, interesting. Yes, these 2 are very hard to distinguish. One of my very faithful volunteers does a remarkable job at restoration painting, In fact, we have stumped the curator a couple times by not being able to tell which is which. But it's C in this case, and again, there's our yellow band of paint, and on there we show the histology project number and the specimen identification. So before we jump to the grinder, I wanted to show you one more thing.
You have just 10 minutes, just to let you know.
10 minutes, just to know. Okay, very good. Here are some drawings, and this is another step that we do that's very important before we begin the histology process. So we'll take a photograph of the specimen, we'll do drawings of the whole bone, and then we'll do individual drawings of the pieces that we take out. And as you can see, we also have letters on these drawings to mark out every single individual segment of the bone. So this is also very important for research over time. So researchers can have the exact measurements from the specimen and then put all the parts and pieces back together in such a way that when they study this, they can orient precisely where that thin section slide came from relative to the whole bone. Okay, so let's go to the grinder.
All right. So I'm going to switch over and show you a video.
Oh, actually, we're going to the saw first.
This video is going to be of Ellen cutting up the dinosaur bone. Just started it already, so if you want to talk, we'll
Okay.
play.
Excellent. And John's going to extract the tibia. There we go.
Okay.
Okay. So you can see I've got a lot of safety gear on. I have earplugs. I have a face mask. I have a face shield. And my bone is braced with medical tape to a board while I'm cutting. Because I've got that very fast moving blade and that very sharp diamond edge, which is doing a wonderful job cutting the bone, but I certainly don't want it to cut me. And then at the end, you're going to see a little grab happen. And that's going to show you why it's important to have your specimen very well braced when you're on the tile saw.
Great. All right.
So I'll bring this with me.
So we followed Ellen over to the other side of the lab. And I'm going to zoom in just a little bit over there. So you can see what she's doing.
Okay. So again, I'm going to start with some safety gear. Angie, am I all in the zone here?
You are, yeah.
Okay,
Good. Yeah.
excellent. So this is the grinder. It powers on. I'm going to turn on the water, and that provides lubrication for the grinding process. I'm going to take my specimen, which now you've seen a few of. So this is the mounted Tyrannosaurus rex bone on the glass plate. I'm going to wet the specimen. I'm going to hit run. I'm going to increase my RPMs, or revolutions per minute, of the plate, or what's called the platen. And then I'm going to actually hold the specimen down. And this takes some practice. You have to keep everything very even. The grinding paper, especially the early stages of it, are very sharp, so you want to wear gloves to protect your fingers. And then we can use a micrometer as we're processing. And a micrometer is something that very, very precisely measures the thinness or the thickness of something. So we can actually chart the progress from this very thick specimen down to that very thin one, which we said at the end will be thin as a human hair. So if I come over here, Angie, can you see what that looks like? Great. So then the next stage would be to come over here to the polisher and use liquid polishing compounds. Sometimes these are also made from diamond, and then that helps get all the scratches out. We get a really shiny, polished surface. It looks nice, and it's important that none of those scratches impair what we're looking at under the microscope or start to look like a structure. So that's a quick view of grinding and polishing. Now I'm going to come over here. And I think what we'll do, I'm going to quickly show you some photo micrographs. So these are pictures of dinosaur bone. You've seen this one already underneath the microscope. this one, also with these beautiful colors. Again, this was the structure I talked about, the osteon, and now I'm looking at you.
There you go.
Very good. This is that line of arrested growth where the dinosaur stopped and then it started growing again. This is the openings where the blood supply came into the bone. And then this photomicrograph, which is definitely one of my favorite, is covered with all of these little black dots. They probably look like ants or pepper granules. So can you make a guess what might have been inside of each of those black dots when the animal was alive? So I'm going to have you think about that.
So we just asked what you think might be inside of those black dots when that animal was alive. What would have been in there?
Were you unable to hear me because of the microphone? Okay, well, we're going to go over all of these terminology again with Maiasaura. So I'll wait for guesses on the dots. Very important structure of bone. And then to note, so far we've looked at adult dinosaurs. Now we're going to shift our gear down to baby dinosaurs. So this is a leg, a rear leg of a baby Maiasaura. And if you were to look inside of this leg, or even one a little bit smaller, at a hatchling stage just out of the egg, this is what you would find. This structure here is called calcified cartilage, and this orange band is primary bone, just beginning to form. So this is sort of captured a moment in time. Bone structure just beginning to form in the end of the leg of that baby dinosaur. And this helped us learn a lot about behavior in this type of dinosaur, as well as what's called physiology, how the whole body runs all of its systems. So now we're going to switch back over and you're going to get to see how that dinosaur bone grows from this stage all the way up to adult.
Would you like me to pull up those last images up here?
Yes.
So you have just a couple minutes.
Okay. I think we can do it. Any questions coming in or guesses on the black dots?
Not yet.
Okay.
Okay.
Okay. So before I move ahead, then I'll just talk to you about, because I'm gonna actually have you look as we go through these slides for those little dots, because they're very, very important. They're actually where the individual bone cells resided during the life of the animal. And we can actually count those, and it gives us an indication about how active the bone was growing and remodeling. So right now you are looking at another hatchling baby dinosaur with lots of that calcified cartilage. So this is what the end of their bones would look like right when they hatched out of the egg. And then the next slide, this is a closer up look at higher magnification. Now again, this shows you that bone forming in the middle around all the cartilage. So you might want to think about what's the difference between cartilage and bone. I bet some of you might even have some ideas about that. And what this helped us understand was because the ends of these dinosaur bones had so much cartilage and so little bone that these baby dinosaurs would have had to stay in the nest and mature further, and would have required parental care for them to be taken care of, because they couldn't have gone out and found food on their own yet. So that really changed the way we saw dinosaurs, and we realized, hey, they're nesting a lot more like birds than reptiles. They have large nesting grounds, they're coming year after year to the same location, they dig nests, they lay eggs, they hatch them out, they stay with their young, and they stay in family groups. So these family groups enabled us to look at the bones of all of the members of the family group, all the way from a hatchling to a nestling. And next we're going to see what a nestling looks like, so the bone's starting to mature. Next we're going to see a juvenile, so that'd be sort of like a middle schooler dinosaur. So we've got less blood supply, but still it's pretty actively growing bone. Next we're going to see a subadult, and you can see a big change here, how the bone got very dense. Next, we're going to see an adult bone, and those openings again, now you're familiar with them, that's the blood supply into the bone, and the bone's starting to do something called remodel. So, the next slide, and we'll skip over that one and go to the end stage. So this is mature bone. So either you're going to see those lines of arrested growth in mature bone, or you're going to see this structure. So the last slide, and then one more. Okay, so all of the structures that we mentioned are in this last picture. And then what I'm going to encourage you to do is I'm going to have Angie go one more slide. And I'd like you to, again, visit our Histology Lab website and go to the page called Worldwide. And you can zoom in to this Google map of the Earth and go to any of those pins and click on them and see what kind of specimens we may have sectioned from localities all around the world. So if you're in a different country today, you can zoom in and see if we've been sectioned any specimens from where you live. And now you can start to think about, are there questions that you have about dinosaurs that you may be able to answer by using paleohistology and looking inside of the bone?
Great.
So any final questions in this last minute?
We don't have any final questions. Well, I'm going to stop sharing this so we can see our scientists again.
Okay, our scientists.
Yay! Yeah. So I'm going to jump back over there and see if you have any more questions. You're welcome to stay on and we'll answer those. But thank you so much for joining us today.
Thank you, everyone.
Yeah, we're glad you got to see us. I'm going to go see if you've got more questions.
Excellent.
All right, stay safe. Thank you so much.
You're so welcome.
Thank
Thank
you. Thank you. They said it's really interesting. They've been doing microscope slides of plants, and this is amazing.
Fantastic.
Yeah. Well, thank you both for tuning in. Have a good rest of your day.
Okay, bye-bye.